Hello, welcome to my poster. I'm Johanna Pispa from the group of Carina Holmberg at the University of Helsinki, and the subject of my poster is what temperature does to the UPS, the ubiquitin proteasome system. So we C. elegans researchers, we grow our worms either at 20 degrees or 25 degrees, and what we started wondering about is whether this 25 degrees actually has an effect on the proteasome. The way we did this experiment is shown in the schematic drawing on the left, which is that we took L4 larvae that had been grown at 20 degrees and placed some of those at 25, and then one day later, when they were about 1 day old adults, we did the comparison using different assays. Now you may wonder whether some kind of stress responses are activated, as we all know that 25 degrees is a bit stressful for the animals. So first of all, we checked for the classical heat shock response, and we did this first by using the HSP 16.2 GFP transgenic line, which is shown in the upper row of the fluorescent images. And the answer is no, we saw no activation using this reporter. On the right side of each of these rows is the positive control. However, when we looked with qPCR at the actual mRNA levels of HSP 16.2, there was some increase, about 7-fold, although this was clearly less than what we could see by doing a heat shock at 32 degrees. Next, we looked for ER stress using the HSP4 GFP transgenic line, and this is the middle panel of the fluorescent images. And here we could see that ER stress was induced. Approximately, the signal increased about 3-fold. And finally, we looked for mitochondrial stress by using the HSP6 GFP line, which is in the bottom row. And here we saw no induction in the intensity of the reporter. We then wanted to check for UPS activity in vivo. And the way we did this is to use transgenic lines which express a fluorescent protein, dendra-2, that has been linked to a non-cleavable ubiquitin. The way these experiments are done is so that the worms are originally green, which is the top row in both of these panels. And by using UV light, we can actually photoconvert part of the green dendra-2 protein into a red form, and this is shown in the middle rows. Then we have a follow-up time, which in the case of the intestinal lines is 6 hours, and in the case of the muscle lines, 24 hours. And then we re-image again. And whatever loss there is of the red signal is a measure of the degradation rate of the reporter. And in this case, we could see that the intestine, there is less reporter left at 25 degrees than at 20 degrees, indicating that the reporter's relative degradation rate is over 30% more at 25 degrees. However, in the muscle, in the body wall muscle tissue, we could see no such difference. In this slide, we've looked at polyuncinated proteins in vivo. And the way we've done this is to use transgenic lines that express the fluorescent protein Z-green, and that's fused with ubiquitin binding motifs and also a degradation signal. In the upper panel, we've expressed this transgene in intestinal tissue, and there's no difference in the intensity of the signal between 20 and 25 degrees. In the bottom panel is the situation in the muscle, which is different, because there 25 degrees on the right induces approximately fourfold increase in the intensity of the transgenic reporter. And this, of course, is in line with our previous results. In the intestine, the UPS activity is increased, and consequently no accumulation of polyunguinate proteins occurs after 1 day at 25 degrees. Whereas in the muscle tissue, there's no change in UPS activity, and this may result in the accumulation of polyunguinated proteins. So last, we looked at the proteasome function in vitro using whole animal lysates. So on the left, we've performed an activity assay for the proteasome, which is an in-gel assay, a native gel, where the gel is bathed with a fluorescent substrate for the chymotrypsin-like activity of the proteasome. This is one of the 3 different enzymatic activities that the proteasome has. And there's no difference between the signal at 20 degrees and 25 degrees. On the right side, in the middle, we have a Western blot analysis where the antibody recognizes 6 of the alpha subunits of the core particle of the proteasome. Again, no difference in the amount of the proteasome seen in the immunoblot. And at the very right, we have another Western analysis where we looked at polyubiquinated proteins. These can be seen as a smear, and again, there's no significant difference between the 2 conditions tested. So the take-home message for this poster is that yes, 25 degrees does make an impact on the UPS. This occurs in a tissue-specific manner, and this is something that should be taken into account when we compare experiments that have been performed at 20 and 25 degrees. And a second take-home message is that when we do in vitro analysis of whole animal lysates, we may sometimes miss the very subtle differences that can be seen on the tissue level. Thank you.